Hello, thank you for joining us once again for Dr. Graves' case studies. This is Mr. Basser, Mr. Yanez, and Ms. Amaya bringing you 16 questions. To start off, I will begin. Figure 1 describes A, B, C, and D, A, B, C, and O-type populations. List the A-type populations, B-type populations, and C-type populations and O-type populations. The article states that population types differ in their generation cycle. What is meant by generation? Generation cycle means the length of time required to reproduce. So here they are. The A-type is given 24 hours to lay their eggs at day 10. B-type is given 2 hours after anesthetization with CO2 on day 14. C-type is given yeast and 48 hours to lay eggs on day 28. And O-type is given yeast and 48 hours to lay eggs on day 70.
Moving on to question 2. Figure 1b shows a phylogeny. In this phylogeny, what do the lengths of the color lines represent? The colors represent the different generation type groups, differing by the time and days started or allowed to reproduce. A phylogeny is shown in figure 1b. The length of the lines represent the number of generations that each population has experienced since they diverged from their ancestor. This means that the longer the branch, the more the population has undergone more generations. In this experiment, A, B, C, and O types are derived populations which all have different generation cycle lengths, such as 10, 14, 28, and 70 days.
Okay, staying on the topic of figure 1, the most recent common ancestor of the five ACO populations, we gathered it was O-type. The most recent common ancestor of the O, NCO, AO, BO, CO, and ACO and B populations, we gathered it to be the O-type as well. All lineages ultimately trace back to the original laboratory ancestral population at the far left of the phylogeny. Therefore, the most recent common ancestor of all these populations is the original livestock population at generation O.
Number 4, based on figure 1b, is the following statement true or false? While the CO populations are on the 28th day generation cycle, most common ancestors were on 14-day generation cycles. We find that this is false. The most recent common ancestor of the CO was on a 70-day generation cycle.
Number 5, figure 1e shows the age-specific fitness of the C-type populations. What is KX? What and why is age-specific fitness near age 28 days in these populations? K of X equals L of X, M of X, where the L of X represents the probability of survival to an age, and the M of X is the fecundity at that age. K of X is the age-specific reproductive rate. The age-related fitness is around 28 days, as this is the average between extended and short breeding times. Shorter breeding times may be too early in reproductive fitness, while longer than 28 days may be too old. The reason that the age-specific fitness is so high at 28 days in these populations is because this is when the flies can reproduce more and contribute more to the population. This ties back to the K of X because it means that the variable L of X is higher for these Drosophila at this age, and the variable M of X, which is the number of offspring they can produce, or the fecundity, is also greatest at this age.
Okay, moving along to figure 3. Figure 3 shows the heterozygosity of each genetic position in the genomes of the newly derived and long-standing A, B, and C population types. So this graph ranges from 0 to 0.5 because the heterozygosity should not be greater than 0.5 in allele frequency. Thus, the y-axis should not be greater than 0.5 as well. Tying it back to Hardy-Weinberg, because the maximum value of 2pq occurs when p is equal to q, when q is equal to 0.5.
On figure 4, we see results comparing different types of populations to each other genomically. For example, the middle panel compares single nucleotide polymorphisms, or SNPs, frequencies of the A-type populations to the C-types at position in the genome. What do the red horizontal lines represent in this figure, and what do the black dots represent? We basically discovered that the red lines represent statistically significant thresholds. The black dots represent the negative log p values of the GWAs, or genome-wide analyses. Each black dot is an SNP plotted by its chromosomal position. It compared the allele frequencies between two population types.
Next question, based on figure 4. Is the following statement true or false? The B and C type populations are more similar to each other than either of them is to the type A populations. We said that this was true. The negative log p values are lower and more similar compared to A versus B or A versus C.
For figure 4 and 5, we compared and contrast what was similar and different. So we gathered that the similarities are both analyzed genomic differentiation between population types, and they both show SNP differentiation across the genome. The differences are that figure 5 seems to have way smaller p-values compared to figure 4. The frequencies are much lower in figure 5 than in figure 4. Ultimately, while the methods are similar, figure 5 seems to have a greater variability when comparing the red line and less intense or lower p-values.
Okay, for question 10, how is table 1 related to figure 4 and figure 5? Explain what the number 10,109 represents in table 1. 10,109 taken to the log results in a log of 4. When compared against an O background in table 5, we see that the compared values do not reach 400, while in figure 4 they do. 10,109 represents the number of SNPs significantly differentiated between A versus C populations.
Next question. Based on table 1, is the following statement true or false? Using 10 replicate populations of each type versus using only 5 replicate populations of each type, you did lower statistical power to identify regions of significant differentiation. We said that this was false. Using 5 replicate populations made variability in the results lesser.
For figure 6, it shows the decay of lineage disequilibrium LD with distance and base pairs away from the focal SNP. So what is a focal SNP? A focal SNP is defined as an evaluation of a single nucleotide that can have multiple implementations or forms. In the case of the genes, there can be multiple types or multiple functions for the same genotype. This focus simply means that the nucleotide is focused in a particular region of the genome. The 3 panels in the figure represent different age groups or reproductive fitness tested, and the colors in each panel represent different population types, such as A type, B type, and C type.
For question 13, in figure 6, we saw that linkage disequilibrium decreases with distance from the focal SNP. What causes this? Linkage disequilibrium decreases as alleles diffuse throughout the genome. When recombination happens between the SNPs, the linkage disequilibrium also decreases. There are few alleles, meaning that there's a high degree of crossing over through the generations. Recombination causes linkage disequilibrium to decrease.
Okay. The author states that they expected linkage disequilibrium to be lowest in the A-type populations due to higher number of generations, which should have allowed for more recombination to occur. However, figure 6 shows that despite this higher number of generations, the A-type populations actually have the highest levels of linkage disequilibrium, and that this pattern is true on all 3 chromosomes shown. Provide a possible explanation for this counterintuitive observation. We said that linkage disequilibrium would be highest in those populations that are forced to mate first. With short reproductive periods, errors are likely to force concentrations of genes to stay in certain spots. These are the highly stressed populations. There could have also been non-random mating, which would cause the linkage disequilibrium to decrease in that population. Type A could have also been a more beneficial trait, which would cause it to be more prominent in the populations over time.
For 15, this question is a multiple choice question. The question reads, this paper uses experimental evolution in the diploid eukaryote, whereas the paper you read for case study 2 uses bacteria. Thinking about the likelihood of adaptive alleles going to fixation and reviewing Dr. Graves' papers, which of the following statements is to be true? We chose B. B reads, in both prokaryotes and eukaryotes, fast adaptation results from new mutations that go to fixation.
On 16, make a list of concepts that appear in this paper that we learned about in class as well. Genome-wide analysis or sequencing, QTL, genetic loading, recombination, heterozygosity, phylogenies, and Hardy-Weinberg principles are all aspects of this paper.
And to answer these last few questions, what was the main research question in this study? The main research question of the study was, what are the genomic foundations of adaptation in sexual populations? What were the main results of this study? The main results were there was a lack of differentiation from long fitness character versus short fitness character. And what were the main conclusions of the study? The populations in nature do not undergo stable selection regimens for 1,000 generations, and thus extreme and unusual evolutionary histories to test competing hypotheses.
Thank you for listening to our little spiel once again on Graves case studies, number 3. Stay tuned for number 4, our final Graves study analysis. Have a wonderful weekend and happy Thanksgiving. Goodbye.